Microbial Matters, a podcast introducing the latest microbiome research. Hi, I'm Jerry Zhang, a chemical biologist in microbiome. In this podcast, I'm going to share with you newest advances in microbiome research, especially those related to human health. Today, I'm going to talk about a paper published in Cell in August 2022, titled Personalized Microbiome-Driven Effects of Non-Nutritive Sweeteners on Human Glucose Tolerance. This work was primarily done by labs of Aaron Segal and Aaron Elinav from Weizmann Institute of Science in Israel. The two labs of the two Aarons have long focused their research on how gut microbiome may impact human health, and they usually involve clinical trials with healthy volunteers. So their results have direct implication on how gut microbiota might correlate with human health. Just to highlight a few research that have caught the headlines of the media, one of them is the glycemic effect of different carbohydrate foods. The conclusion is that the so-called high glycemic index or low glycemic index foods are not absolute, and the blood sugar response of each individual is different. For example, some individual would have faster glucose level rise when eating whole wheat bread than eating white bread, which is the opposite of common perception. Another one of the research studies shows how antibiotics may impact human gut microbiome and whether taking probiotics might help the recovery of the disrupted microbiome. And the result is astonishing in that the commercial probiotics on the market at the time are actually delaying the recovery of human gut microbiome after antibiotic treatment. Back to the paper that we're discussing today, labs of Aarons want to study whether the mainstream sweeteners on the market might impact human health and whether that is related to gut microbiota. The sweeteners included are saccharin, aspartame, sucralose, and stevia. In the United States, these are pretty easily distinguished because they have different colors on their package. Sugar will be in white package, saccharin in pink as sweet and low, aspartame in blue as equal, sucralose in yellow as Splenda, and stevioglycoside in light green as stevia. I'm from southern China and I have sweet teeth since childhood, so I love to add sugar sweetening packs to coffee. And I also drink a lot of diet drinks like Diet Coke and Coke Zero. And after a while, I get to tell apart which sweetener I'm drinking. And I feel like sweetness of saccharin is the flattest. And I can tell this artificial sweetness with first sip. Aspartame is better, but still flat and straight. It tastes like soap without MSG. You know, quoting Uncle Roger, "Hiya, MSG is the king of flavor." It always fascinates me that aspartame is, in essence, a dipeptide, a compound composed of two amino acids. So the fact that it tastes like sugar but not protein is just amazing. Stevia has a sweetness with more layers, and its aftertaste is a little bit herbal, like traditional Chinese medicine, which is not surprising because it is originated from a plant in the daisy family. My favorite is sucralose or Splenda because I simply cannot tell it apart from sucrose, the sugar. But since COVID, I don't add sugar or sweeteners to my drink anymore, but that's another story. The catabolism of these sweeteners in human body is also different. Sucralose is hard to be absorbed, so most of what you eat are excreted in poops. Saccharin can be absorbed slowly in the gut, and only about 10% will be in the poo as it is. Stevia is known to be catabolized by gut microbiota because it contains glycosagonds. Aspartame is quickly absorbed in the upper GI tract because it's a dipeptide. These sweeteners are labeled 0 calories, not only because they cannot be turned into calories by humans, they're also super sweet compared to sugar, so that it takes only a tiny teeny bit to reach your desired sweetness, and it's less than 5 calories, which can be labeled as 0 calories according to the FDA. But that tiny bit is too small and hard to use, because if you get a shiver when you were adding sweeteners to your cake dough, you would get a cake too sweet to eat. So these sweeteners would blend with a bit of sugar and be blown up during processing, so that you feel you're adding similar volume of sugar when you open each small pack. Back to the article discussed today. To study the impact of sweeteners on human health, Aaron's labs recruited 1,375 healthy human volunteers for screening, and they hoped to find those who had never eaten sweeteners, knowingly or unknowingly, so that they can rule out the prior exposure to sweeteners would have already affected the gut microbiome and health and interfered with this experiment. After screening, only 131 volunteers were eligible. So you can tell that the vast majority of people have already eaten sweeteners. And I believe this is the case in most industrialized countries. These volunteers were divided into 6 groups of 20 people each, and 4 of them were given 1 serving of one of the sweeteners per day for 2 weeks. And the dose did not exceed the daily intake recommended by the FDA. As mentioned earlier, because the sweetener packet also contains sugar, in order to eliminate the interference of sugar, the 5th group ate the same amount of glucose as in the sweetener packet every day, and the last group didn't eat sugar or sweetener as in control. During 2 weeks, the researchers collected stool samples, blood samples, etc. from the volunteers, who had also worn real-time blood glucose monitors on them. In addition, as each individual shown internal control, samples were collected 1 week before and 1 week after the sugar or sweetener consumption experiment. Everyone has already done a glucose tolerance test before, during, and after the experiment, which is to drink a glass of sugar water to measure blood sugar fluctuations. The greater the fluctuation, the worse the glucose tolerance. I'll just say the main conclusion of this article. The first major conclusion, which surprised me a lot, was that the volunteers who ate saccharin and sucralose had worse glucose tolerance during the 2 weeks they ate the sweetener, and it was worse in the 2nd week than in the 1st week. Conversely, eating aspartame or stevia or even glucose had no such effect. Unfortunately, after they stopped eating sweeteners, their glucose tolerance levels returned. In other words, eating saccharin and sucralose can temporarily make people's ability to regulate blood sugar worse. The second major conclusion of the article is that sweeteners will change the gut microbiota, and changes in the microbiome in response to sweeteners are also different from person to person. For example, in the experimental group that ate sucralose, not everyone's glucose tolerance level became worse. Some people just didn't change at all. The researchers selected 4 volunteers who responded the most to sweeteners and 3 volunteers who responded the least, transferred their gut microbiome to germ-free mice, and then tested the glucose tolerance of these mice. It was found that the response in humans can be well reproduced in mice, which shows that the gut microbiome does indeed mediate the changes in glucose tolerance. Then the authors also specifically studied the impact of each sweetener on the intestinal microbiome and the changes in the metabolic functions of the microbiota. I will not go into details, and all in all, it varies from person to person. Although the conclusion of this article is very eye-catching and there are some new discoveries, I think there are still some flaws. For example, because the experiment recruited volunteers living in a developed country, like Israel, who had never eaten sweeteners, they may be eating very simple diet, so their gut microbiota may be very different from those of ordinary people. it's hard to say whether the results obtained in this way are applicable to the general population. Secondly, there are only 20 people in each group in the experiment, and the number of volunteers is still too small, so the conclusion obtained from the experiment may be biased due to small sample size. Finally, I feel that the impact of sweeteners on human health is still being studied actively, and some articles maybe have exaggerated the harm of sweeteners, blending with pseudoscientific opinions. Moreover, the author of this article also emphasized that the conclusion of these sweeteners should not be interpreted as eating more sugar is better, because the harm of eating too much sugar has long been demonstrated by a large number of studies. After all, it's human nature to love sweets. For patients with metabolic diseases, sweeteners can indeed help them satisfy their desire to eat sweet without consuming sugar. The affluence of life allows us to eat more sweets, but as an old Chinese saying goes, too far is as bad as not enough. Even if the sweet is using sweeteners with zero sugar and 0 calories, everyone should eat them in moderation and live a healthy lifestyle. Cool, that's it for today. Thanks for listening. You can listen to more episodes in your favorite podcast platform like Spotify, Google Podcasts, and Apple Podcasts. You can also leave your comments. It'll really help my podcast. You can also support my podcast, Microbial Matters, on Anchor. Thanks again.